Public health officials and pharmaceutical industry representatives are facing a significant challenge following reports that existing vaccine candidates may not provide adequate protection against a newly identified mutation of the hantavirus. This admission marks a critical juncture in the global effort to manage rodent-borne pathogens as the medical community grapples with the inherent volatility of RNA viruses. For those unfamiliar with the underlying science, hantaviruses represent a family of viruses spread mainly by rodents. They are responsible for diverse clinical presentations ranging from hantavirus pulmonary syndrome, or HPS, which is prevalent in the Americas, to hemorrhagic fever with renal syndrome, or HFRS, which is more commonly seen in Europe and Asia. The announcement regarding the lack of vaccine efficacy is particularly concerning because hantavirus infections carry high mortality rates, and the medical community has long sought a standardized, globally available preventative measure. The current situation highlights a gap between laboratory development and the rapid pace of viral evolution. While hantaviruses are not typically known for the same high-speed transmission rates as respiratory viruses like influenza, their ability to mutate within their rodent reservoirs creates a persistent moving target for genomic researchers. The framing of this development as a significant setback by major pharmaceutical entities underscores a broader reality in virology, that a single genetic shift in the virus's protein structure can render years of research and stockpiled biological materials effectively obsolete. Viewers should understand that this is not a statement of total defeat, but rather a frank acknowledgement of the current limitations in medical countermeasures. The geography of this concern is widespread, as hantaviruses are endemic to various regions where human activity overlaps with rodent habitats. As rural expansion and environmental shifts continue, the frequency of human-rodent contact increases, making the failure of a vaccine stopgap a matter of significant public health urgency. In the absence of a biological shield, the focus must necessarily shift back to the fundamental mechanics of the virus itself. This includes understanding the specific glycoprotein mutations that have allowed this strain to bypass the immune responses previously triggered by experimental or existing vaccine formulations. By acknowledging these shortcomings now, health agencies are signaling a shift in strategy, prioritizing the identification of the mutation's reach and the development of more robust, broad-spectrum therapeutic options. The core of the current development lies in the technical verification processes conducted by specialized laboratories and the subsequent statements from industry stakeholders. Pharmaceutical researchers have indicated that the specific mutated strain in question demonstrates significant antigenic drift, a process where the virus's surface markers change enough that the immune system no longer recognizes it based on previous exposure or vaccination. This admission follows rigorous testing where existing serum and vaccine-induced antibodies were introduced to the mutated virus in controlled environments, only to find that the neutralizing effect was substantially diminished or absent. While specific names of labs or clinics are often withheld for proprietary or security reasons in the early stages of such reporting, the consensus among major medical manufacturers is that the current technological pipeline requires a fundamental reset. This is a complex logistical and scientific hurdle. Developing a vaccine involves identifying stable regions of the viral genome, but the hantavirus is notoriously difficult to culture and study in high-volume settings. The industry's admission serves as a contrast to earlier, more optimistic projections regarding the readiness of hantavirus countermeasures. It highlights a recurring theme in global health reporting, the tension between the slow, methodical pace of clinical trials and the unpredictable nature of zoonotic spillover. Furthermore, the role of surveillance agencies is now under the microscope. To understand the scale of this mutation, public health departments must rely on genomic sequencing from human cases and rodent samples collected in the field. This data is then fed back to pharmaceutical entities to inform the next generation of vaccines. However, the current lack of protection means that for the immediate future, clinical facilities are restricted to providing supportive care rather than preventative immunization. There is no evidence in the source material to suggest that this mutation affects the mode of transmission, which remains primarily through the inhalation of aerosolized excreta from infected rodents. However, the pharmaceutical admission clarifies that for those who do come into contact with the virus, the anticipated safety net of a vaccine is not currently an option. This has immediate implications for logistics and shipping in regions where workers may be exposed to rodent-infested environments, as previous safety protocols may have relied on the assumption of eventual medical intervention that is now being called into question by the very companies tasked with producing it. Given the current landscape where pharmaceutical interventions are limited, public health guidance remains the most effective tool for prevention and containment. Health agencies emphasize that the primary defense against hantavirus, mutated or otherwise, is the rigorous control of rodent populations and the minimization of human exposure to their habitats. Surveillance programs are being urged to increase the frequency of environmental sampling to track the geographic spread of the mutated strain. Practical prevention involves sealing entry points in homes and workplaces, using professional-grade disinfectants when cleaning areas where rodents have been present, and ensuring proper ventilation in confined spaces like sheds, cabins, or basements. It is vital to avoid stirring up dust in these areas, as the virus is most commonly contracted through the inhalation of tiny particles. Regarding the limits of current knowledge, it remains unclear exactly how widespread this mutation has become, or if it significantly alters the severity of the disease. Science is an iterative process, and while the current admission from pharmaceutical companies is a setback, it also provides a clear roadmap for what needs to be addressed in the next phase of research. There is no reason for panic, but there is a clear need for heightened awareness among those in high-risk occupations or environments. The global health community continues to monitor reports from clinics and laboratories to determine if the clinical presentation of the mutated strain differs from the classic symptoms of fever, muscle aches, and respiratory distress. Until a new generation of vaccines can be formulated, tested, and distributed, the responsibility for safety lies in individual and community-level mitigation strategies. This involves a transparent dialogue between officials and the public regarding what is known and what remains under investigation. The honest assessment of these biological limitations is a hallmark of a functioning public health system, allowing for the reallocation of resources to where they are most needed, namely, diagnostic accuracy and patient support. As the situation evolves, the focus will remain on the intersection of virology, ecology, and human behavior. We will continue to follow these developments as more data becomes available from official health channels. For more deep dives into global health surveillance and reporting, consider subscribing to this channel. The core of this technical setback involves a fundamental shift in the viral architecture of the hantavirus, specifically within the glycoproteins that reside on the outer envelope of the pathogen. To understand why current vaccine candidates are failing, one must look at the laboratory procedures used to evaluate viral resistance. In high-containment biosafety facilities, researchers utilize a metric known as a neutralization assay, where antibodies generated by a vaccine are introduced to the virus to see if they can effectively prevent it from infecting target cells. The recent admission from the pharmaceutical sector indicates that these antibodies are failing to recognize the mutated strain, essentially allowing the virus to bypass the primary line of immunological defense. This phenomenon, often referred to as antigenic drift, occurs when the virus's genetic material, specifically the M segment which encodes the GN and GC proteins, undergoes mutations that alter the physical shape of these surface structures. Because vaccines are typically designed to train the human immune system to recognize a very specific molecular footprint on the virus, any significant structural change can render the entire preventative strategy ineffective. Now, this development forces a significant pivot in how public health agencies and private laboratories approach the problem. Instead of proceeding toward large-scale manufacturing or clinical distribution, research teams must now return to the genomic sequencing phase. This involves utilizing next-generation sequencing to map the exact location of the mutations and determine if they are consistent across different geographic clusters or if the virus is continuing to diversify. The industry's acknowledgement of Z failure marks a significant moment of transparency regarding the limitations of current biotechnological platforms. It underscores the difficulty of creating a universal hantavirus vaccine, a goal that has eluded scientists for decades due to the high degree of genetic diversity among the various known strains, such as Sin nombre in North America or the Hantan and Pumala viruses in Eurasia. For clinical staff and hospitals on the front lines, The lack of an effective vaccine means that the treatment protocol remains strictly reactive rather than proactive. In areas where the mutated strain is suspected to be circulating, diagnostic laboratories are being urged to update their primer sets for polymerase chain reaction, or PCR, tests to ensure they can even detect the new variant with high sensitivity. There is an inherent lag time between the identification of a genetic mutation and the recalibration of diagnostic tools, which creates a window of uncertainty for patient management and epidemiological tracking. Furthermore, the pharmaceutical industry's report suggests that the existing stockpiles of experimental doses may now hold limited value for the specific regions where this mutation has taken hold. This creates a logistical vacuum, as the focus of global health resources must shift from potential immunization campaigns to enhanced environmental surveillance and case tracking. While the source material does not specify a fixed timeline for the development of a revised vaccine, historical precedents in virology suggest that a complete redesign of the viral vector or the mRNA sequence could take months or even years of rigorous clinical testing to ensure both safety and efficacy against the new structural profile of the pathogen. This reality places a significant burden on state and international health monitors to provide accurate, real-time data on the prevalence of the mutation, as any further geographic spread could complicate the efforts of laboratories already struggling to keep pace with the virus's natural evolution within its rodent reservoirs. With the pharmaceutical sector acknowledging that a medical solution is not currently available for the mutated hantavirus strain, the burden of protection shifts decisively toward preventative environmental management and rigorous public health surveillance. The absence of a vaccine means that the primary defense remains the interruption of the transmission cycle between rodent reservoirs and human hosts. For individuals living or working in areas where hantaviruses are endemic, the practical takeaways are centered on the concept of rodent exclusion and safe decontamination. Public health agencies advise that the most effective way to prevent infection is to eliminate rodent access to homes, cabins, and storage facilities. This involves sealing even the smallest gaps, some no larger than a quarter-inch, with durable materials like steel wool or metal flashing, as hantavirus-carrying rodents can compress their bodies to enter remarkably tight spaces. Furthermore, it is critical to address attractants by securing food sources, including pet food and birdseed, in heavy-duty, chew-proof containers. These measures are not merely suggestions, but are the current front line of defense in a landscape where biological countermeasures have failed to keep pace with viral evolution. When cleaning areas where rodents have been present, the methodology is a matter of life and death due to the risk of aerosolization. The virus is shed through the urine, droppings, and saliva of infected rodents, and it can remain infectious in the environment for varying periods depending on temperature and humidity. The central guidance from health officials is to never use dry methods like sweeping or vacuuming in contaminated areas, as these actions kick microscopic viral particles into the air where they can be easily inhaled. Instead, the area must be thoroughly wetted down with a disinfectant or a solution of 1 part bleach to 9 parts water. After soaking the area for several minutes, the debris should be removed using paper towels or rags while wearing protective gear, such as gloves and, in high-risk scenarios, N95 respirators. These protocols are designed to keep the virus grounded and inactive, significantly reducing the probability of human infection. Surveillance efforts are also being intensified, with field biologists and epidemiologists monitoring rodent populations to determine if the mutation has altered the virus's prevalence within the host species. This ecological data is vital for predicting potential hotspots and issuing timely warnings to the public. There remain significant limits to the current understanding of this mutation, which public health experts acknowledge with professional caution. It is currently unknown whether this genetic shift has impacted the virulence of the pathogen, meaning whether it causes more or less severe disease than previous strains, or if it has altered the incubation period, which typically ranges from 1 to 8 weeks. Ethical reporting requires acknowledging these gaps in knowledge rather than filling them with speculation. The integrity of the public health response depends on this transparency. By admitting that current vaccines are ineffective, the industry is prioritizing factual accuracy over a false sense of security. This ethical stance allows for a recalibration of resources toward the development of better diagnostic tools and the expansion of clinical supportive care capacity. As we look forward, the situation serves as a stark reminder of the dynamic nature of zoonotic diseases and the continuous need for global cooperation in viral monitoring. The scientific community is now tasked with building a more resilient, broad-spectrum response that can better withstand the inevitable mutations of the natural world. While the news of vaccine failure is a setback, it reinforces the necessity of grounded, evidence-based prevention and the importance of maintaining high standards of environmental hygiene. We will continue to track official statements and lab reports as the global health community works to close this gap in protection. For more in-depth reporting on emerging public health challenges, consider subscribing to this channel.